Good morning students, hope you are all well. Okay, now we'll continue the lesson organization of tissues. In this lesson you are going to study about the cell division. Now cell division, majorly pointed out that cells are formed from the pre-existing cells, and the cell divisions is divided into three types: amitosis, mitosis and meiosis. Okay, amitosis means direct division, then mitosis means indirect division, and meiosis means reduction division. Now we'll see the amitosis. Now you can see the cell, and cell is surrounded by cytoplasm, and in the center you can see the nucleus. The nucleus is surrounded by nucleolus. Now if you come to the cell, the nucleus is present in the center. First the nucleus will elongate and constriction appears in the middle. Okay, this is the constriction. Constriction appears in the middle. So along with the nucleus, the cytoplasm also divides. So the nucleus divides into two, and the cytoplasm also will divides along with the nucleus. Then the nucleus will form two cells. As a result, they will form two daughter cells. The next one is the mitosis. Okay, mitosis. If you come to the mitosis, you can see the parent. That parent will divide into two identical daughters, and it was discovered by Fleming. Okay, and these two identical daughters, and they will have the nucleus. The nucleus will have the same amount of DNA, chromosomes and genes. Okay, so the two daughter cells will have the same amount of DNA, chromosomes and gene as a parent. So this process is also called as an equational division. And in the mitosis you can see the two events. The one is the karyokinesis. The another one is cytokinesis. Okay, this event means the nucleus will divide. In cytokinesis means cytoplasm will divide. So in this mitosis you will see two events, and there will undergo four phases: prophase 1, metaphase 1, anaphase 1 and telophase 1. Now here the interphase, the nucleus will take rest. So it is the resting place of the nucleus. So that's why they are called as an interphase. The next one, the chromosomes will become very short and thick. That is seen in the nucleus. The centrosomes splits into two daughter centrioles. The centrioles move towards the opposite poles. The centrioles are surrounded by radiating rays, Astor rays. Spindle fiber appears between the centrioles. Nuclear membrane and nucleolus disappear. Then the next one is the metaphase. Here you can see the duplicating chromosomes is present in the equator plane and form the metaphase plate. Chromosomes are attached to the spindle fibers, and it is known as chromosomal fibers. In chromosomes, the centromere divides into two with the chromatid. The next one is the anaphase. Here the centromere with the chromatids divides and chromosomes will move towards the opposite poles. The daughter chromosomes achieved by the contraction of spindle fibers. Okay, the next one is the telophase 1. The daughter chromosomes lengthens and form a thinner chromatin threads. Then spindle fiber breaks. This is the spindle fiber. Here you can't see the spindle fiber because the spindle fiber breaks and disappear. The nucleus and the nucleolus membrane reappear in the daughter cells. The next one is the cytokinesis. Here the construction occurs in the middle. The nucleus deepens and divides. The cytoplasm also divides. So the nucleus also divides and cytoplasm. And as a result, they form two daughter cells from the parent. The next one is the importance of mitosis. Okay, in the first point, the daughter cells. In the daughter cells will receive the same amount of chromosomes, genes and DNA material. So, in the daughter cells, first point, the daughter cells will receive the same amount of DNA, genetic material and chromosomes. Then in second point, the multicellular, only in multicellular cells, they will have the growth in the organs, tissues and in the cells. So, they will increase as the body weight. Then third point, they will repair the wounded and damaged tissues. The last point is they will replace as the old and the dead cell. So, it is the important 2 marks question. Meiosis was coined by Farmer. Okay, and here the division will take place sex cells or gametes, male or the female gametes. Now chromosomes. The chromosomal number reduced to haploid from diploid, and meiosis will form the 4 daughter cells. Meiosis will have 2 divisions, heterotypic division and homotypic division. The heterotypic divisions will undergo 4 phases: prophase, metaphase, anaphase and telophase. And prophase will undergo 4 stages: leptotin, zygotin, pachotin and diplotin. Okay, first we will see the prophase. Here the chromosomes is uncoiled and become thread-like structure in nucleus. Then in zygotin, the homologous chromosomes become pairing, and this pairing is called as an synapsis. Then in pachotin, what happens? The chromosomes are very visible and 2 pairs will form the bivalent, and it contains the chromatids. Homologous chromosomes, each pair separate but remain X-shaped. The chromatids crossing over take place and the genetic recombinations take place. The next one is the diplotin. Chiasmata will move towards the chromosomes and form the centromere, and this is known as terminalization. Then diagenesis. Here the paired chromosomes will become very short, thick and disappear. The nuclear and the nuclear membrane will disappear. Spindle fibers will make the appearance. In metaphase 1, the chromosomes move towards the equator. 2 chromatids do not separate. Then in anaphase, the homologous chromosomes with the chromatids undivided and move towards the opposite poles, and this stage is known as diet. And in the telophase, the haploid number of chromosomes become uncoiled and elongates. The nucleus and the nuclear membrane reappear. Two daughter nucleus are formed. The last one is the cytokinesis. The cytoplasmic division occur. As a result, two haploid cells are formed. Cytokinesis. Here the cytoplasmic division occurs and they will form two haploid cells. So, as a result, they will form two haploid cells. Significance of meiosis. Constant number of chromosomes is maintained in the meiosis division. Here the crossing over taking place. So, because of that, you can see the characters will be changed from one generation to the another generation. Okay, important 2 marks question. The next one is difference between mitosis and meiosis. The mitosis will take place in somatic cells. Somatic cells means daughter cells. But meiosis will take place in the reproductive cells. Here the growth continues throughout their life, but the growth is the homotypic division. Here two haploid cells divide. As a result, 4 haploid cells are formed. They will undergo 5 stages: prophase, metaphase, anaphase, telophase and cytokinesis. Now we will see the prophase 2. Centrioles divide into 2 and they will move towards the opposite poles. As does spindle fibers appear. Nucleus and nuclear membrane disappear. The next one is the anaphase 2. Two daughter chromosomes move towards the opposite poles. Daughter chromosomes move towards the opposite poles. The next one, telophase 2. Daughter chromosomes are centered, nuclear membrane and nucleus appear. Then cytokinesis. 2 cells are formed from haploid daughter cells. As a result, 4 cells with the haploid number of chromosomes are formed. More information about the cells. Age of our body cells. The cells of islands, nerve cells, muscle cells. Muscle cells lost a lifetime but once dead are not replaced. Epithelial cells lining the gut last only about 5 days. Average life of other gut cells is about 15 years. Duration of cell replacement. Skin cells about every 2 weeks. Bone cells is about every 10 years. Cells about every 300 to 500 days. This is liver cells. Stem cells are undifferentiated cells which undergo unlimited division and give rise to one or more different types of cells. Embryonic stem cells differentiate into different tissues and organs. Stem cells are used in the treatment of degenerative disease. In present days, umbilical cord blood is collected at the time of childbirth and it is stored in stem cell banks to treat any diseases in the future.